Greetings everyone, and today we are going to discuss our life cycle of HIV in detail. So, starting with the first stage of life cycle, as previously introduced to its first step, which is the binding stage or adsorption or attachment stage. What actually happens in this stage is that the CD4 receptor of the host cell, CD4 receptor of the host cell basically infects CD4 cells. So, CD4 receptor of the host cell attaches with the GP120 glycoprotein spike of the HIV virus, and this attachment acts like a lock and key model, such that when this lock and key model is attached and a complex is formed, that complex tends to move away. As the complex tends to turn aside, it causes the HIV virus to have its lipid bilayer fused with the host cell's lipid bilayer, but that includes the second stage, which is the fusion stage. First and foremost, when the complex is formed, binding occurs, attachment occurs. As there is mutual affinity of rubisco with carbon dioxide and oxygen, same way there's mutual affinity of GP120 glycoprotein spike with CD4 cell. And after that fusion occurs, what is fusion in this process? As the binding adsorption has occurred, this binding leads the lipid bilayer of the host cell to tremble, to start to shiver because of the co-receptors present. Co-receptors have once the receptors are attached with the glycoprotein spikes, the co-receptors are also in contact with the glycoprotein spikes. As co-receptors are in contact with the glycoprotein spike, they become activated, as a result of which it leads to fusion of the host lipid bilayer with HIV envelope, as a result of which, uh, as there is first and foremost activation, after which there would be a trembling started within the lipid bilayer, due to which it will change its structure and start to tremble. Once it is done with it, it will now bind with the lipid bilayer of HIV, which means the envelope of HIV will be attached with the lipid bilayer of host cell, as a result of which, when it will bind together, this process will be known as fusion. And once fusion has occurred, the HIV will be entered within the cell, just as uh, a phagocytosis process occurs. Now, as HIV has entered within the host cell, the third step which would be occurring over here would be the reverse transcription. The diagram has a bit changed from what is being taught in our books. So here, as you can see, there is process of reverse transcription, in which the uncoating of HIV will occur. Uncoating of HIV will occur by the enzyme which is known as protease. Protease is an enzyme which works in uncoating as well as in maturation of HIV, as well as during the process of cleavage of proteins when it assembles its structure into the immature HIV virus particle. So in this case here, protease acts and degrades the protein shell. As it degrades the protein shell, rest of the viral genome as well as the viral enzyme reverse transcriptase, integrase, and protease are then released within the cytoplasm. Now at that moment when it is released within the cytoplasm, there the RNA, a viral genome of HIV, is quite unstable. Why? Because it is RNA, ribonucleotide. It is having hydroxyl group on its second carbon, due to which it is having more mutations, less stability as compared to DNA. DNA is a more stable particle. Moreover, RNA is having a characteristic that whenever RNA is entered within the cell, there's an enzyme which is present in the cell cytoplasm known as RNase, which can degrade the viral uh, which can degrade the viral genome. So, in order to protect it, there are several viral proteins which also protect the viral genome from the RNase enzyme, as well as, even though in our in eukaryotic translation, there's also sometimes formation of heterogeneous immature mRNA, which directly goes inside the process of translation for protein synthesis rather than forming mature mRNA. This is because there is the presence of mRNA present within the nucleus, that in order to run away from the RNase enzyme present within the nucleus, the heterogeneous immature mRNA tends to run away from it and directly enters inside the translation process within the cytoplasm, so that the heterogeneous mRNA cannot get infected by, I mean, not, of course, not get infected, but not get degraded by the RNase enzyme, because RNase enzyme has a capability to degrade RNA, due to which mRNA, heterogeneous immature mRNA, tends to run away from the nucleus and directly take part in the translation process for protein synthesis. Why? So that it cannot get degraded by the RNase enzyme. Once again, here the same condition is taking place now, as the viral genome and viral enzymes are present over here. This process of RNA is now converted into RNA is now converted into DNA. Now RNA, now there is formation of reverse transcription here. A DNA strand, a complementary DNA strand known as cDNA strand, is made from the RNA strand, which is from 5 prime to 3 prime, as a result of which the complementary cDNA strand which would be formed would be from 3 prime to 5 prime. As I mentioned in the previous video as well, that here there is a 5 prime to 3 prime strand, one is, which is the RNA strand, and on another strand which is present over here is known as the 3 prime to 5 prime strand. So as you can see, this is a strand which is present over here. Now this 5 prime to 3 prime strand and 3 prime to 5 prime strand is formed, I mean 5 prime to 3 prime strand acts as a template strand for 3 prime to 5 prime cDNA strand, which is done by the addition of deoxyribonucleotides with the help of an enzyme which is known as RNA dependent DNA polymerase reverse transcriptase, or RTase. Now, as a result of this, from the RNA which acts as a template strand, the cDNA is formed. Now, when cDNA is formed, there is a linkage formed between both of them, which is due to the hydrogen bonding. Now, as hydrogen bonding is formed between both of them, here there is the presence of a new molecule which is known as the DNA RNA hybrid molecule. This is a DNA RNA hybrid molecule. Now, in order to separate the DNA RNA hybrid molecule, the reverse transcriptase enzyme also works as RNase enzyme, such that it breaks down the DNA RNA hybrid molecule's hydrogen bonding, due to which RNA 5 prime to 3 prime is separated on one hand side, while cDNA is separated on another hand side, and 3 prime to 5 prime cDNA now creates its complementary strand, as a result of which cDNA acts as a template strand for the other strand of cDNA. Now in this case, again the same enzyme reverse transcriptase will be utilized, but this time it will act as DNA dependent DNA polymerase rather than RNA dependent DNA polymerase, as a result of which there would be a complementary strand form which would be from 5 prime to 3 prime. Again, cDNA acts as a template strand for another strand of cDNA, which means complementary DNA of complementary DNA acts as complementary DNA of RNA acts as a template strand for complementary DNA of the complementary DNA. So in this case, this is the same condition over here as well. One more thing, that once the double-stranded DNA is formed within the cytoplasm, the process of reverse transcription is done, and now this double-stranded DNA, which is a stabilized form, will be now sent within the nucleus for the process of integration. Now, when it will go inside the host cell nucleus, it will integrate with the host cell genome. How? It will utilize the enzyme integrase. Integrase is an enzyme which first and foremost has two capability: first, integration of host genome with viral genome; second, of cutting the host genome so that it can incorporate the viral genome into the host genome. So when integrase enzyme acts, it causes to cut the host genome, as a result of which, when integrase enzyme cuts the host genome, it can also be known as restriction. It can also perform the action of restriction endonuclease, not the original endonuclease but restriction endonuclease, due to which it cuts the host genome but cannot uh, entirely degrade the host genome, just like it does in just like the endonuclease enzyme does in the bacteriophage. Here, it only acts as restriction endonuclease, cuts the host genome, and incorporates the viral genome, as a result of which this integrase enzyme also works as restriction endonuclease. As a result of which, once the integrated DNA is formed, this is known as provirus. Now, this integrated DNA is known as provirus or prophage. Now, once a provirus or prophage is formed, it starts to replicate itself, form its copies. Now, uh, virus after integration the will only form a few copies of it for 2 to 3 weeks, but since it will not find any favorable circumstances, it will tend to remain silent after that period, such that it will remain silent for around 8 to 10 years, or maybe more than or less than that. After this period, in which, first and foremost, this period in which it will remain silent is known as the latency period, or dormant period. While on the other hand side, the period after which it will be awakened period in which it would be awakened will be considered as the dangerous outcome of its disease, known as AIDS, acquired immunodeficiency syndrome, which will act out as a dangerous disease. So in this case, it can cause infection, but initially there is no replication. There is replication, but of only of a few copies, after which it does not make its copy and enter the dormant period. Now, as we consider that it has entered inside its dormant period, latent period, it stays within it. No new copies are formed, or few copies are formed. Now, after that, there is transcription process occurring. Transcription is a process in which the genes present within the viral genome tend to code the mRNA, which is responsible for the formation, which is responsible for acting as either viral genome, or responsible for acting in the process of translation to form the viral protein, so that it can create its copies. Now, once this structure forms, there is a requirement of an enzyme known as RNA polymerase, which is a host enzyme. Once RNA polymerase is utilized, mRNA is formed, then viral genome and viral protein are formed accordingly, either through translation or by directly using mRNA as viral genome, as a result of which the structures are formed. The HIV codes its viral proteins, which are known as polyproteins, which are large proteins. So in order to break these large proteins in the stage of assembly, there is requirement of the enzyme protease, in order to cleave the large polyproteins into smaller proteinaceous subunits. Once these smaller proteinaceous subunits are formed, by the way, why does the HIV remains silent for many years? This is because it has a gene regulatory system. As living organisms are having gene regulatory system, same way HIV is also having a gene regulatory system, in which it utilizes its inhibitor. It utilizes its inhibitor to turn the genes of replication off, as a result of which the genes are unable to perform their function. Now, uh, after that, as the genes are turned off, it remains latent for for much of the time, and then it attacks, and then as viral genome and viral RNA, viral genome and viral proteins are formed. Now cleavage occurs for protease enzyme, after which it causes to assemble the viral genome with viral protein, due to which an immature HIV particle is formed. Now during the maturation, there is also work of protease, as well as once immature HIV particle is formed. During the process of budding. Budding is a process in which the viral gene, in which the immature HIV borrows a lipid bilayer from the lipid bi borrows a lipid bilayer from the host cell. As a result of which, when it borrows a lipid bilayer from the host cell, it causes the lipid bilayer of the host cell to decrease, and gradually, as many of the viruses are particles are formed, it causes a lipid bilayer of the viruses to lipid bilayer of the host cell to decrease by the virus gradually, such that there is a time coming when the viruses are having um, the host cell's lipid bilayer completely shortened, due to which it either becomes bursted, or it becomes destroyed, or it just becomes non-functional. Now, when they borrow the lipid bilayer, there's a process of exocytosis, through which they perform exocytosis, and then they are they form a bud first and foremost, from where they do exocytosis, and then they are released as mature HIV particles known as mature virions, as it may be seen in diagram as well. It may be seen in the diagram as well. Moreover, when it comes to the mature HIV virion, it then further infects other cells alongside bursting or making the infected cell non-functional. So as a result of this function, it causes to complete the HIV cycle. Now, HIV cycle started with infection, and it start ended at infection as well, which means it is showcasing the characteristics of cycle, right? As a result of which, one more interesting fact, that if we drink the solution of HIV's viral genome only, no enzymes, no proteinaceous structure, no lipid bilayer, no glycoproteinaceous, only viral genome are drinking, then there won't be any effect of that viral genome over our health, even though we are openly drinking it, because the viral genome alone cannot do anything. Even though if it enters the cell through binding and fusion, and somehow just manages to enter the cell within the cytoplasm, the RNase enzyme present will start to degrade it. And even though if it is not doing reverse transcription and directly entering within the translation step, due to which this phenomenon is known as RNA interference. Even though if it tries to do RNA interference directly undergoing translation, it will not be able to do so, because in translation there is an enzyme RNase present, which causes to degrade the viral genome. So one more thing, that even though if RNase enzyme is also not present, then there would be um an instability present within the viral genome. It is very unstable particle, due to which it can not survive for more than half an hour, due to which it will degrade itself and not be able to infect ourselves. So as an adaptability, HIV virus requires reverse transcription. Its gene are coded for reverse transcription, because reverse transcription is something which makes it adaptable to the situation of uh, causing such a dangerous infection, as well as surviving within the host cell. One more thing, that here HIV virus is also not having HIV virus is only able to infect the host cell uh, because of the fusion. I mean, here the fusion of the HIV virus with the host cell is only possible if there are co-receptors and receptors of CD4 cells present. These CD4 receptors and its co-receptors are essential for infection of HIV virus. That's all about the video. Hopefully, we'll meet again in another one.